Welcome to ISL Enabled Virtual Lab of CSIR.
And now it's time to ask a scientist.
Welcome back, everybody. Now we have Dr. Megha Kumar, a senior scientist at CCMB Hyderabad. Among many other exciting things, she studies about how animals, zebrafish embryos, develop and how errors during cell division causes embryos to develop defectively. So, Jigyasa, let's go ahead and ask our questions, shall we?
So, is it possible that if we put this regulator in other animals, like let's say a whale or a cat or a dog or any other animal like that, is it possible that they will also not develop limbs?
Yes. So if we look at example by the kind of animal you're talking about. So if we consider, say, whale, it doesn't have limbs, right? It's swimming like fish in the water. So if you look at the embryo of a whale, you see a similar pattern just as the snake, that the limb genes are present in the embryo, but this regulatory element or this regulator is absent. And that's why the whales don't develop the limbs. But if you look at a cat or a dog, they have fully formed limbs, right? The hind and the forelimbs. So if you look at a dog or a cat embryo, yes, this regulator is present and that results in formation of the limb. So if we take a snake regulator and put in a cat embryo, yes, you will see some sort of reduction in the formation of the entire limb. So the hind limb or the forelimb will not be completely formed because this regulator is going to stop that activity. So evolution has not gone in a linear or a very straight manner. But what has happened is multiple times during evolution, different kind of animals have lost different set of genes.
And how do you know it is only this gene that is responsible for whatever you see in the embryo?
So what different scientists have done is try to tinker and manipulate different genes and see what kind of effects they have in the embryo. So sometimes some genes can directly affect a process. Sometimes there are combination of genes that work.
How are you interested in asking such questions? Like, what brings you to ask so many curious questions about embryos?
So why am I interested in this field is because I feel embryos are spectacular biological entities which are so dynamic and they change constantly, right? It starts with one fertilized cell where male and female cells fuse. So the sperm and the oocyte. And then the embryo has all the information stacked up inside it to form a complete individual, right? And how these cells start to divide, how do they know that they are neighbors to this cell? And how do they know their orientation? And how they form different structures? How do the cells communicate to each other?
Did you always know that you wanted to study this?
So till high school, till class 12, I liked all aspects of biology. Okay. But when I came to undergraduate during my bachelor's is when I studied developmental biology as a subject where you look at different embryos. Okay. So in undergraduate is when I realized that this subject is very interesting and it gave me a basic primer on how the embryo tends to develop over time and how different structures form. Becoming a scientist will give me a full-time, full-day chance to look at embryos, to study, to do any kind of experimentation or ask questions that I'm interested in. So then I decided to pursue science as a career for research. During my PhD, I started working with fish and they are very nice, small, 2 to 4 centimeter long fish, the zebrafish. And they're called zebrafish because they have stripes like the zebra animal does. So that's why we named it zebrafish. And now we understand that more than 80 to 90% of the human genes are also present in the zebrafish. So most diseases that happen in the humans can be checked in the zebrafish. So if you look at a zebrafish embryo, it's actually transparent. So you can see different structures forming in real time just by using a simple microscope because it's transparent. And at some point, I'm studying basic biology for the interest of my curiosity, but also to understand what kind of defects happen in children when they are born with such problems, right? So then that gives you a clinical aspect of understanding how embryos develop and why babies have certain problems and defects when they are born. So only when we understand basic science, we'll be able to take it to understand what kind of defects happen in human babies.
So this has been amazing, and we really enjoyed knowing that it's possible to see transparent fish embryos in the microscope and see them develop. So if somebody is as curious as you and is determined to find answers to their curious question, what is that kind of advice would you give to our listeners in that context?
I think find something that you really, really enjoy doing and you are passionate about. Those two things have to come, that you have to be passionate about it consistently and enjoy what you're doing, and choose a profession that makes you do these two things together. So you may not have to be a scientist to enjoy science, right? There are many other kinds of careers you can pursue after doing basic science also, but choose a profession where you're really, really enjoying it and you're passionate. And that's when you become something like Sachin Tendulkar of your field. Okay. So you don't have to be doing cricket to be Sachin Tendulkar. You can be Sachin in your comfort zone, right? And that's the most important thing.
Thank you so much.
Do you now understand why snakes don't have limbs?
Yes, it's a long story spanning millions of years. Researchers say that some snakes of the past had a mutation in a gene which tells their body to develop limbs. As a result of this mutation, these snakes did not develop limbs. But all these snakes without limbs somehow not only survived but also thrived. So eventually, all snakes of the present generation had this mutation and they continue to pass it on.
Exactly. And how do scientists know that the mutation was only in this particular gene?
Scientists like Dr. Megha Kumar study model embryos to understand the roles of different genes in embryo development.
Yes, and Dr. Megha studies one such model in her lab. While she enjoys pursuing her curious questions in the lab, she advises our listeners to find what brings them joy and then pursue it with all their passion. But listeners, what we know about snake embryos and snake genes may change over time.
Who knows? One of our listeners might study snakes and find out something about them that we never knew before. So, that's it for today.
If you want to know more about today's scientist, check out their profile. The link is in the show notes.
If you have any questions you want us to explore, shoot them away to indiaaskswhy@gmail.com.
If you would like to talk to us directly, then join the fun on our Telegram group. Again, the link is in the show notes.
For updates on India Asks Why, follow us at India Asks Why on Twitter and at India_Asks Why on Instagram.
Shweta N. Hegde and Ruchi Manglunia are the hosts for the podcast.
Indulekha MS edited this episode.
And Khushi Goyal transcribed it.
And we are funded by India Biosciences' second outreach grant.
Until next time, till then, stay tuned and stay curious.